Normal cell can be transformed by a different number of mechanisms. It can be exogenous, external mechanism, any external toxin or any infection like any viral infection. You know, DNA viruses, they can also cause cancer. Any toxic metabolite that is derived from the body, like say reactive oxygen species. They have the capacity to damage DNA. Basically what happens is the DNA is damaged, right? And the ability of the cell to repair this damaged cell or repair the DNA is lost. And now the cell has the capacity to grow in an unlimited manner. It has unlimited replicating potential. There are multiple mutations that take place, not a single mutation, multiple mutations take place, and then the cell is transformed into a malignant cell. Okay, so these chemicals, there are some chemicals like say aniline dyes, right? They can cause bladder cancer. Smoking, nicotine can cause lung cancer. Everyone knows this, everyone ignores it. And apart from that, ultraviolet radiation, they can cause melanoma. You all know that sun exposure can cause melanoma. So the DNA damage lies at the heart of carcinogenesis, right? And the defective cell now can multiply in an unregulated manner and cause distant metastasis and all the effects of cancer. This is what I have told. So it can be environmental, like this. These are the environmental, or it can be any genetic or germline mutations. So can you tell me of any genetic mutation that can cause cancer? Any gene which is very important for cancer development? Yes, HER2 new. Apart from that? P53? Okay, so they can be genetic or germline mutations which can also be inherited. Apart from that, it can be spontaneous or fall into the category of bad luck. Anybody can develop cancer. It is coming up in a big way. If you want jobs later on in life, you will have to understand this. This is a big field for jobs. Everyone is going into, after you specialize, everyone is going into cancer. Poishao beshi, kajo pabe. Right? So, this I have already explained. A tumor is formed by the clonal expansion of a single precursor cell that has incurred genetic damage. So, one particular mutation starts the whole chapter. One particular mutation that is found in all the cell. After that, other mutations the cell adds on to that particular mutation. Can you understand? So, one particular mutation, say this is the normal cell, it has, say P53 was damaged, first P53, or say RB gene. RB gene is also very commonly mutated in most cancers. Mainly it is found in retinoblastoma. RB, it was first found in retinoblastoma, so RB, but it is actually found in most cancers. So the RB gene was say damaged and then doubling, and then the RB gene defect will be found in all the cells. It can be point mutation or copy number variations. So can you tell me of any copy number variation that can be diagnosed by fish? CML? Do you know what Philadelphia chromosome? So that is also that one particular mutation will be found in all genes, and when we diagnose also, the part of pathology is we diagnose the disease, so we have to look for that. Say we have to diagnose CML. It is all molecular. Nobody looks under the microscope and diagnoses anything anymore. So it will be diagnosed by fish or cytogenetic analysis, all this. So the genes, the 4 classes of genes are principal targets of cancer-causing mutations. Growth-promoting proto-oncogenes, growth-inhibiting tumor suppressor genes, genes that regulate programmed cell death or apoptosis, and genes that are responsible for DNA repair. Right, these four classes of genes are the main genes which can be mutated and cause cancer. Carcinogenesis results from the accumulation of complementary mutations in a stepwise fashion over time. So this is what I was talking about. There is one single mutation, say the first mutation, the important mutation, that is called driver mutation. That is important. It gives rise to the hallmark of cancer. It can be diagnosed and it gives the cancers a particular characteristic. Say RB gene, that's mutated, it gives rise to retinoblastoma or osteosarcoma. Now, say this cell is actually growing in the eye only. It is not yet metastasizing. So, to metastasize, it needs another mutation. So, these important mutations, for angiogenesis, there's going to be another mutation. So, these important mutations are called driver mutations. Apart from that, there are passenger mutations. Right. The passenger mutations are other mutations that are actually accumulated by the cell. But these mutations are not phenotypically active. They are not expressed, but they are also found in all the cells. And the passenger mutations accumulate to give a particular characteristic to the cancer. Say prostate cancer. Prostate cancer preferentially metastasizes to the spine. Or even breast cancer, they preferentially metastasize to bones. So these the cancer needs these driver mutations and passenger mutations to get these specific hallmarks. They behave, every cancer behaves in a specific manner. So that these are called driver mutations and passenger mutations. So what happens? This is the initiator mutation, say, which gives rise to DNA instability. The initiator mutation, I told you, it is the mutation present in all the subsequent cells. Then additional mutations which give rise to genomic instability. Then there will be additional driver mutations which will give rise to the characteristic of, say, angiogenesis, metastasis, etc. And then, so what happens is the tumor is growing. And like Darwin suggested, when he went to, I think, Galapagos Island, and he studied finches. Yeah. So he suggested that there is selection, right? The survival of the fittest. So it works here also. Survival of the fittest. Not all cancer cells will mutate. Not all cancer cells will spread. Particular cells will spread, and those cells are selected. So here also survival of the fittest. Not all cancer cells will metastasize. Even when we see under microscopes actually, say I have a tumor from colon, right? It looks a certain kind. But when it has, say, metastasized to the omentum. The omental metastasize look a little different even under light microscope. So not all cells have the ability to metastasize. Particular cells can. So that is the selection. This cell was actually selected. It was able to metastasize, spread, kill the patient. So this picture is from Robbins. The normal cell, carcinogen-induced mutation. Then there are additional driver mutations and accumulation of passenger mutation, additional mutations, subclones have emerged, and this is heterogeneous. Not all look same. Some cells look certain kind, other cells look certain kind. Say there's something called, when you will study systemic pathology, you will see there is something called signet ring cancer, poorly differentiated adenocarcinoma. Signet, it actually looks like a ring. A signet ring, that's why it's called signet ring cancer. It's very common. So not all cells have signet ring feature. To diagnose something as a signet ring cell adenocarcinoma, it's very common. We need at least 50% of the cells must have that signet ring feature. So not all cells that I see will have the signet ring feature. Only some will have. So there are emergence of subclones. Or say a neuroendocrine carcinoma. Say there can be something like, say, an adenocarcinoma with neuroendocrine features. Neuroendocrine features mean some hormones are selected which give rise to some additional symptoms, right? So not all cells will have the neuroendocrine features. Some cells will have. This we can actually diagnose under light microscope. So it's relevant when you ultimately diagnose. So now we come to the main thing, cellular and molecular hallmarks of cancer. I will discuss these, right? Let me go into it. Self-sufficiency in growth signals. Now, any cell, you know, require growth signals to grow. External growth signals are required. But the cancer cell does not require any external growth signals. It can grow on its own. So that's self-sufficiency in growth signals. So what happens is there are proto-oncogenes, you know, the proto-oncogenes, like say the JAK-STAT pathway. It's very common in all myelogenous leukemias, right? Chronic myeloid leukemias, the JAK-STAT pathway is mutated, right? Apart from that, say the EGF receptor pathway. EGF receptor pathway is also very important for lung cancer. Okay, then the RAS pathway for melanomas. Or the HER2 news, as you said, HER2 pathway for breast cancer. So these are proto-oncogenes. The normal cell has these proto-oncogenes. Now it's mutated. So it becomes an activated oncogene. The activated oncogene can actually replicate on its own. So what are oncogenes? They are mutated or overexpressed versions of normal cellular genes. The normal cellular genes are the proto-oncogenes. These are mutated and give rise to oncogenes. The oncogenes secrete oncoproteins. The oncoproteins are freed from the normal checkpoints. Normal checkpoints that are regulated by P53 and all. The normal checkpoints, it can disregard the normal checkpoints and proliferate. Next, insensitivity to growth inhibition. That is what is done by the tumor suppressor genes, like P53, like RB. RB gene for retinoblastoma, osteosarcoma. P50, there is a syndrome. Li-Fraumani syndrome. APC, hereditary adenomatous polyposis coli. It gives rise to adenocarcinoma. Have you seen House MD? Dekhecho kyo? There was an episode. People who have inherited APC, they have to undergo, even if they, because 70% chance they will develop adenocarcinoma of colon. Multiple colonic polyps will be found, which will undergo transformation to adenocarcinoma. It is 100% in, say, 35 to 40. So before, I think 25 years of age, before they attain that age, they have to go colectomy. This was actually shown in one episode. Now, NF1, neurofibroma. NF2, schwannoma. BRCA1, BRCA2, you know, it's very famous gene. Why is it famous? Yes. Why was it in the news? Do you know? Some famous person inherited BRCA gene. Angelina Jolie. She underwent ovariectomy. Okay, STK11. WT1 for Wilms tumor. This one is for Peutz-Jeghers, MSH2, MLH1, MSH6. This one you don't have to know. It's for hereditary non-polyposis colonic cancer, and VHL, it gives rise to renal carcinoma, von Hippel-Lindau syndrome. Right, so the tumor suppressor genes, now they actually prevent the cell from basically crashing. They prevent the cells from excessively proliferating. So there are two tumor suppressor genes, right? Say there are two homologous chromosomes. Now, this is the normal gene. Say there is the first mutation. It can be inherited, it can be acquired. The first mutation. Even then, the cell will not become a malignant cell. It has to have two hits. This is called Knudsen two-hit hypothesis, first diagnosed for retinoblastoma. So for inherited retinoblastoma, one gene is probably mutated in utero, and within the first few years of life, the child, inevitably, the other retinoblastoma gene is mutated and develops retinoblastoma, unilateral or bilateral. So this leads to cancer. Okay, now this I find very interesting. Growth promoting metabolic alterations. Who is he? He is Otto von Warburg. Okay, what did he give us? He told us about this pathway, aerobic glycolysis. He discovered this and he won the Nobel Prize for it. Right? So what is this? Normally what happens? Either oxidative phosphorylation, where from one molecule of glucose, 36 molecules of ATP are manufactured. Krebs cycle, or anaerobic glycolysis, right? When there is less oxygen in anaerobic conditions, one molecule of glucose gives rise to 2 molecules of ATP. But cancer cells, but not just cancer cells, any cell that can proliferate, like say any embryo, the embryogenic cells, they also show similar metabolic pathway, which is called aerobic glycolysis, where 4 molecules, even in the presence of oxygen, this like a glycolytic pathway, anaerobic glycolysis, takes place in the absence of oxygen. But here, even in the presence of oxygen, cancer cells prefer this pathway where 4 molecules of ATP are produced from 1 molecule of glucose. This is important in a very popular test for diagnosing malignancy and metastasis. Do you know? Have you heard of PET scan? Yes. Yes. So F18 fluorodeoxygenated glucose, that is used, right? And that actually uses this Warburg effect only. This is selectively taken, and any cell which is taking up that F18 labeled glucose, yes, F18 labeled glucose, it actually shows in the tomography, in the scan. So we can diagnose the metastasis, right? Even proliferating tissue like bone marrow, that will also uptake this. That will also light up. So the bone marrow will light up, any other metastasis will light up. So what happens is, this kind of altered metabolism, when tumor cells have the ability to show this kind of altered metabolism. So why? Now why is the tumor cell doing this? Why not go for the easy pathway like the Krebs cycle, where you get 36 molecules of glucose and not just 4 molecules? It needs energy. Any cell needs energy. So this prefers, because it gets some carbon moiety from this. The cell, not only it needs DNA, it needs to manufacture the whole cell, all the organelles. It needs protein, it needs fat, all the cell organelles need to be manufactured. So for that, the Warburg effect is preferred by the tumor cells, so that they get the carbon moiety and they can manufacture the other organelles required for the cell to grow. Apart from that, there is something called autophagy. This is another regulating pathway where, say, a cell cannot grow. The ideal conditions are not there. It may be DNA damage, it may be something else. Now, the cell can ingest its own organelles. Now, cancer cells, this is also something interesting. Cancer cells can either evade autophagy, or they can use autophagy to become dormant. Now, it's obvious why they are evading autophagy. They need to grow. They disregard everything else. They just go on growing by autophagy. But they evade autophagy and they just go on growing. But say, person is getting treatment, say any radiation or any other treatment. Now, the cell, what it can do is it can ingest some own organelles and go into a dormant state. So we think what? The person, we have treated the patient is free from cancer, patient is happy. Now what happens? Relapse. After a few years, relapse. Why? This is one pathway. It becomes dormant, the cell is dormant, we have not been able to diagnose it. We think person is cured. But then after 10 years, 5 years, 2 years, 20 years, it's variable for different cancers. This patient again has a relapse because some cells had evaded the treatment, right? So this is one pathway. Apart from that, there is something called oncometabolism. This is seen in some enzymes of Krebs cycle. This enzyme shows mutation, isocitrate dehydrogenase. This is actually very important for brain cancer. To diagnose brain cancer, we need to diagnose this. Now, other thing is evasion of cell death. I have already said that the cell death, it evades. It can occur by loss of TP53 function or overexpression of anti-apoptotic members of the BCL2 family, like say in case of translocation 14-18 leading to follicular lymphoma, anti-apoptotic members of the BCL2 family are mutated. Okay. And then targeted drug therapy against this has been developed to treat this kind of lymphoma. I have already said this also, limitless replicating potential. All cancers contain cells that are immortal and have limitless replicative potential. You have heard of HeLa, Hep2, immortal cell lines. You have heard of them? These are actually, HeLa is from Henrietta Lacks. She was diagnosed with ovarian cancer, and she had donated her cell line for research. These are immortal cell lines which are used for research, for diagnosis, etc. So they have the limit, they never die. They have limitless replicative potential. Now, 3 factors are involved. Evasion of senescence. Senescence means death. Evasion of senescence. Now, normally any cell can actually multiply up to say 60 times or 70 times. After that, it will die. Any normal cell will die after 60 to 70 replications. But say there is a mutation in RBP53 which gives rise to DNA damage and it can evade cell death. Apart from that, there is evasion of mitotic crisis. Now, not all cells, telomerase is required for making telomere. Normally, cells telomere shortens, and once it shortens, after up to a certain degree, the cell will die. Not all cells have telomerase. Only stem cells or embryo cells, they have telomerase. But cancer cells can also have telomerase, and this is another way by which they have limitless replicative potential. And of course, there is the capacity for self-renewal. Like the cancer stem cell, what happens is normally, see a cell is dividing. It will give rise to 2 differentiated cells. But in case of cancer cell, one cell will remain a stem cell, the other will be differentiated. Again, say there are these 2 cells. These 2 cells will give rise to 4 cells. Out of these 4 cells, one will remain a stem cell, the other will be differentiated. Like this, a stem cell is always retained within the cancer so that they can self-renew. This is not normal, not normally found in all cells. Now we come to angiogenesis. It is a very important hallmark of cancer. Now, a tumor cannot grow beyond 1 to 2 millimeter size unless it has the capacity to induce on angiogenesis. So, this small initial tumor, it can only grow up to 1 to 2 millimeters. So, where nutrients from the neighboring vessels will reach. The cancer cell, of course, also requires nutrients to grow. All cells require nutrients to grow. So, the nearby vessel can only supply for a growth up to 1 to 2 millimeter. After that, it will not grow unless it can cause neovascularization, new vessel formation. Neovascularization is a process by which new vessels sprout from capillaries. There are nearby blood vessels and capillaries. Now, the cancer cells, they make the endothelial cells secrete. They induce the endothelial cells to grow nearby and by growth factors like vascular endothelial growth factor, VEGF. So by this growth factor, the blood vessels will grow near the cancer. Now, these endothelial cells will also provide some growth factors like IGF, insulin-derived growth factor, and PDGF, platelet-derived growth factor. These also help the tumor to grow and they supply the nutrients. So, the tumor is benefited by two ways. It is getting growth factors, it's getting nutrients, it can now grow well. It grows, 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 and then it can reach a large enough size so that it can also metastasize. Now, these vessels, the endothelial lining in these vessels or the structure of these vessels is not normal. Not normal like when we have the ones which are formed during embryogenesis or any other, say, there is trauma and then there is vessel growth. These vessels are not like that. These grow in a haphazard manner. These grow in a kind of disarray, and this helps in metastasis. Angiogenesis is an essential facet of malignancy. No tumor can survive unless it can induce angiogenesis. You can get short notes itself on angiogenesis. Now, invasion and metastasis. This is another very, very important hallmark of cancer. Now, what is invasion? It is local spread. What is metastasis? We understand metastasis as distant spread. Now, what happens? Say this is the tumor cell. It is transformed. It is growing, 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 growing. Now, there is a basement membrane. It needs to cross a lot of barriers to cause the metastasis. This cell is growing. It is acquiring other additional driver mutations, as I said. Now, this cell can now pierce the basement membrane. For cell-cell adhesion, there are some molecules. For cell adhesion to basement membrane, there are some other molecules like, say, E-cadherin. Now, E-cadherin is a very important molecule which is mutated in many cancers like lobular breast cancer, gastric adenocarcinoma, and we also have a treatment against this E-cadherin mutation, right? So, E-cadherin helps the tumor to, now this cell will actually penetrate the basement membrane. When we diagnose a cancer, we say are getting a biopsy sample. Now, I am seeing that the cell has grown only here and the basement membrane is intact. I will label it as carcinoma in situ. It's a very important term. It's important for, say, cervical cancer, breast cancer, DCIS, carcinoma in situ. It's always diagnosed. So, it needs to pierce the basement membrane so that I can say that it is actually malignant. Now, the cell has to pierce the basement membrane, traverse through this part, passage through the extracellular matrix, then pierce the basement membrane of the neighboring vessel. Then it will have to form a tumor embolus. Now, this tumor embolus, not all will survive. Not all will cause metastasis. It will need some other additional mutations and multiple mutations, not just one mutation that I can name. Multiple mutations are required so that this tumor embolus can actually go and home to another vascular bed, basically. This is a vascular bed. Like, say, any tumor of the GIT, colon cancer or anything. Now, the draining vascular bed is usually the liver. So, liver is the most common site for metastasis, right? Another is lung. Lung, liver, brain, bone. This is a mnemonic, lung, liver, brain, bone. You have to remember most common site. You cannot remember, teacher asks or your examiner asks you, what is the common site for metastasis? Just say lung, liver, brain, bone. Apart from that, as I said, there are some specific sites. Say for breast cancer, it is bone. For prostate cancer, it is spine. For neuroblastoma, it is, or for lung cancer, it is adrenals. So they have the preferential sites where they can home, right? So the cell actually has to pierce the basement membrane, traverse the extracellular matrix, pierce this basement membrane, form the tumor embolus. The tumor embolus actually is coated by the platelets. Then it has to pierce this and extrude into the vascular bed and then cause distant metastasis. Apart from that, lymph node. Everyone knows about sentinel lymph nodes. Have you heard of sentinel lymph nodes? That is the first lymph node which is affected by metastasis. We have to, when the surgeon sends a cancer to the pathologist, he will separately send the sentinel lymph node so that we can stage the cancer. So that is a part of the metastasis. Now, evasion of immune surveillance. So within the blood, there are even some immune cells, or even when the cancer is growing here, the every cell actually undergoes some kind of immune surveillance, like an antivirus. It actually surveys the entire system constantly. Now, this cell has to evade the immune surveillance. Immune surveillance is actually the effector cell is CTL. Do you know what is CTL? Nah. Cytotoxic T lymphocyte. Yes. CD8 plus cytotoxic T lymphocyte. It acts by the MHC class 1 molecules. So, now the normal function of the immune system is to constantly scan the body for emerging malignant cells and destroy them. Now, the cancer cell has to be such so that it can evade the cytotoxic T lymphocytes, right? Now, normal cell, it has some normal antigens which is expressed with MHC class 1 molecules. You've had classes on this, MHC class 1 molecules. So, the MHC class 1 molecules helps the CTL to recognize that this is normal cell. Normal antigens are expressed, normal cell. Now, how does the CTL recognize a tumor cell? It can be by 3 pathways. It can be either, say the normal protein has now become an oncoprotein. The protein is altered, the MHC class 1 molecule is showing altered protein, CTL recognizes it, kills the cell. Another pathway is the normal proteins are expressed in an excessive manner, which is also recognized by the MHC class 1, CTL recognizes and kills the cell. Other is DNA viruses. Name some DNA viruses that can cause cancer. HPV? HPV causes what? I'm sure many of you have been vaccinated against HPV. Cervarix and Gardasil. These are 2 vaccines which I did not get, but I'm sure you got. These are some vaccines against HPV to prevent cervical cancer. Cervical cancer is the most common cancer among Indian women. Breast cancer is overall, cervical cancer is in our developing nations actually. So HPV is one DNA virus that can cause cancer. Apart from that, EBV, have you heard of EBV? Epstein-Barr virus? Yeah, Burkitt's lymphoma. So these viruses also will, when a cell is affected by these viruses, it will, the MHC class 1 molecule will show some altered protein which is also recognized by CTLs, and the CTL will kill this cell. Now, say if a person is immunocompromised, right? If a person is immunocompromised, they preferentially are affected by these DNA viruses and they have more chances of developing cancer. Say a person is suffering from AIDS, will get Kaposi's sarcoma. You've heard of Kaposi's sarcoma? It actually happens in AIDS patients preferentially. So these DNA viruses can affect the cell. If the person is immunocompromised, CTL is not working, the person gets cancer. Apart from that, the cell can also evade these CTLs. The principal immune mechanism of tumor eradication is killing of tumor cells by CTLs specific for tumor antigens. Now, who are these two? I'm sure you don't know. I also didn't know unless I studied this. These people, they got the Nobel Prize in 2018 for physiology and medicine. His name is James P. Allison, and his name is Takusu Honjo, I think. So they together got the Nobel Prize because they discovered these CTLs which are specific for tumors. Now these CTLs can also be used for therapy. Therapy is being developed to eradicate cancers using these CTLs. Tumors may avoid immune system by either selective outgrowth of antigen negative variants. I already told you about Darwinian survival of the fittest. Now say other cells are eradicated. One cell gets a certain mutation where it can evade the CTL. The cell is not showing, the cell is mutated, but the MHC class 1 molecule is not showing that mutated oncoprotein. The CTL does not recognize it. The cell evades this cytotoxic killing. Other part is, say, reduced expression of the MHC. MHC takei noshto kore dilo. So what happens is, the cell again evades killing by the CTLs. Apart from that, there is immune suppression mediated by tumor cells. Tumor cells also cause immune suppression in the patient so that it can evade killing by the CTLs. Now, genomic instability. Patients with inherited mutations in genes involved in DNA repair systems are at greatly increased risk of developing cancer. She, to remember Braca, I always remember her because she underwent a lot of surgeries to avoid this kind of cancer. Because it is actually diagnosed in those countries. In our country, nothing is diagnosed. Only when the cancer develops, the person dies, oh, acha, cancer hoichilo. That's what basically happens. So now normally P53 arrests cell division to repair any DNA damage. If it cannot repair the DNA damage, it goes apoptosis. Mutation of P53 is a common finding in most cancers. Now there are something called mismatch repair genes. MLH2, MSH6, these are the mismatch repair genes which cause microsatellite, something called microsatellite instability. If you don't understand, forget it. Microsatellite instability which causes hereditary non-polyposis colonic cancer. APC gene causes familial adenomatous polyposis. HNPCC is caused by these mismatch repair genes. There are other inherited DNA repair defects like nucleotide excision repair which causes xeroderma pigmentosum. Now, the patients of xeroderma pigmentosum frequently get melanoma because the radiation causes DNA damage. This is not repaired by the nucleotide excision repair. They have a defect in this. So, DNA damage caused by radiation is not repaired. The patients of xeroderma pigmentosum get melanoma frequently. Apart from that, there is Bloom syndrome, ataxia telangiectasia. These patients get leukemia, lymphoma because they cannot repair the DNA damage present in these cases. Then there is Fanconi's anemia, which also causes leukemia. Apart from that, BRCA1, BRCA2. This is also a part of genomic instability, which causes familial breast cancer. Apart from that, ovarian cancer. Now this is the end. So carcinogenesis is a multi-step process, as I said, driver mutations, additional passenger mutations, other driver mutations. It is a multi-step process. It develops over a long time. Nobody can predict how long cell will be malignant before it actually expresses itself or causes metastasis. Say, even patients of acute lymphoblastic leukemia, ALL. Normally they are diagnosed, say, when the child is about 9 years old or 10 years old. But this child, even 10 years before the child was, say, diagnosed with ALL, we can find the mutated cell which is circulating within the system. But it had not expressed, it had not caused the symptoms of ALL. That is why it wasn't diagnosed. So it is a multi-step process. Lots of mutations the cell will have to acquire a lot of mutations to actually express itself as a malignant cell. The mutations are accumulated in a stepwise fashion, and they act in complementary ways to produce a fully malignant tumor. There is something else called cancer-enabling inflammation. Now, normally a cancer patient will be cachexic. They will come with weight loss. They will come with frequent infections, ability to acquire frequent infections because of these cancer-enabling inflammation. Epigenetic changes like DNA methylation, they also play a role in tumor spread.
So today we will discuss about local anesthetics. As the name suggests, it is the anesthesia is localized. So what is the first thing is what is the difference between the local anesthetics with the general anesthetics? What is the main difference? Local anesthetics, it reduces or removes the pain sensation primarily, and this loss of pain sensation is reversible. Whereas for general anesthetics, there is a loss of consciousness, and that is also reversible one. So main difference between local anesthetics and general anesthetics is the loss of consciousness in case of general anesthetics, and whereas only pain sensation is lost in local anesthetic, even that it is reversible. It is reversible pain sensation, loss of pain sensation by the local anesthetic. So it is effective and reversible block of impulse along the nerve axons that lost due to the primary blockage of sodium gated channels. Sodium gated channels are blocked. This is the primary mechanism, and mainly among all sensations, different sensations. What are the different sensations other than pain? There are vibration sensation, temperature, but the main loss of pain sensation is the main for the local anesthetics. So, what is the chemical nature? There is a aromatic ring which is lipophilic. Then there is a hydrophilic one, that is tertiary amine, and they bind with the bond. That bond may be ester bond or amide bond. So there is a lipophilic portion, there is a hydrophilic portion, and they bind with a two specific bond, either by the esterase bond or amide bond. As ester bond quickly metabolized by butyryl cholinesterase, which is present in the plasma. So the action of ester type of local anesthetics is much less or short duration. Like procaine, it is less than 1 minute. Like that. So there is in chemical nature, there is a lipophilic component, there is a hydrophilic component, and they bind with a bond. That bond may be amide or ester. If it is ester, it metabolized early in the plasma by the butyryl cholinesterase, and so most of the esterase type of local anesthetics are short acting. So first, in the pharmacokinetic, what are the part of the pharmacokinetic? What are the parts? Absorption, distribution, metabolism, and excretion. Very good. So in absorption, there are factors which determine its systemic absorption. Systemic absorption of local anesthetics. Like dose of the drug. If the drug dose is high, absorption will be high. Similarly, site of injection. If it is vascular, the absorption will be high. Next is drug tissue and drug binding. If binding is high, then there is a less free available drug for absorption. So if high drug tissue binding, there will be less absorption. Physiochemical property of the drug, if it is lipophilic, easily absorbable, hydrophilic, less absorbable. Now last one is the use of vasoconstrictor. Use of vasoconstrictor, how it acts? How it acts? Name some vasoconstrictors. Vasoconstrictor in naam ki bolo? Already discussed. Why adrenaline is used with local anesthetics? Adrenaline stimulates alpha-1 receptor which is present in the vascular smooth muscle. They vasoconstrict, so systemic absorption will be less. So presence of the drug, local anesthetic will be, duration will be high. So action of local anesthetic duration will be prolonged when it is used with vasoconstrictors. If it is highly vascular area like tracheal mucosa, it is more absorbed systemically. Poorly perfused organ like tendons, dermis, subcutaneous fat, the blood flow will be less. So systemic absorption will be less. So if we go for the regional anesthesia, the absorption is like highest in intracostal because there is a more vascular one, followed by caudal, epidural, brachial plexus and sciatic nerve. This is the flowchart for the regional anesthesia. So MCQ, which one is fastest absorbing for the local anesthetics? For regional anesthesia, highest will be intercostal, followed by caudal, epidural, brachial plexus and sciatic nerve. Vasoconstrictor already discussed. As vasoconstrictor like adrenaline, they stimulate which receptor? Alpha-1, which is present in the blood vessels. Alpha-1 present in the smooth muscles of blood vessels. It stimulates vasoconstriction. There is vasoconstriction. As there is a vasoconstriction, what happens? The absorption of local anesthetic will be, vasoconstriction hole less or more? Less. So the site of action presence duration will be prolonged. But it is only applied for the intermediate type of, and there are long duration of local anesthetic, not the esterase type of, because they are already very rapidly metabolized, indifferent to presence of vasoconstriction. It is more important for the longer acting or intermediate acting local anesthetic. Distribution, amide widely distributed after even intravenous bolus. Intravenous bolus, they are widely distributed. First, they goes into the highly perfused organ. What are the highly perfused organ? Like brain, like heart. They first go there. Then there is a slower distributional phase where moderately well perfused organ like muscles and GI tissue they present. And there is so first rapidly perfusing organ followed by the moderately perfused organ. But there is another picture, that is sequestration, like fat cells. They can accumulate in the fat cells, and for this is called sequestration. For sequestration, it also can increase the duration of the action. Sequestration of local anesthetics can help for the longer duration of action. Metabolism. Esterase metabolized in plasma due to the butyryl cholinesterase, whereas amide type are metabolized in liver, and it became more soluble, water soluble and easily excretable through the urine. Acidification of the urine promotes the ionization of the tertiary amine base. So it helps to excrete the local anesthetic from the body. Most of the drug, when alkalization of the urine causes increased excretion like barbiturates. But in case of local anesthetic, acidification of the urine. Acidification of urine increases the excretion of the local anesthetics. Now, mechanism of action. Mechanism of action primarily depend on the sodium channel. Now, sodium channel has three states. One is resting state, one is active state, another one is inactive state. In case of resting state, the extracellular phase is blocked, but the inner cellular gate is open. Whereas for activated state, both are open. In case of inactivated state, the extracellular phase is open, but the intracellular phase is blocked. So there are three phases: resting, activated, and inactivated for the sodium channel. So primarily blocking with this local anesthetics to the inner surface causes the effect of the local anesthetics. Resting membrane potential for the nerve is close to minus 90 to minus 60 millivolt. When the impulse came, there is an activation of the sodium channel and sodium will go inside or outside. Go inside the neuronal cell and they goes up to the plus 40. After that, there is a inactive state of this sodium channel. So resting, then activate due to the impulse, then go up, voltage go up to the 40, plus 40, then there is a inactivation. There is an inactivation. In case of inactivation, which gate will be blocked? Intracellular or extracellular? Intracellular is blocked. So in this case, after that it is plus 40. Now there is a opening of the potassium channel and there is a potassium inside the outside, from the inside to the outward, and it goes back to the minus 95. This is the basic principle for the sodium channel to generate the impulse. But due to in the inactive state, local anesthetics bind with the inner surface and block to generate any impulse. So local anesthetic binds the receptor near the intracellular end to the sodium channel, and this is time dependent and voltage dependent fashion. The blockage is time dependent as well as voltage dependent. So the sequence for reducing this impulse, first there will be increased threshold followed by flow of the impulse conduction. Threshold will be high, then impulse conduction will be slow, followed by there will be the potential will be slope will be less stiff, then there is the amplitude will be decreased. So first there is a resting membrane potential will be high, then there is a slow conduction, then the slope will be less stiff, then there is a decreased amplitude of the action potential, and after all this there is the abolition of the conduction. So now this is the 5 step of the blockage of conduction. So if there is a myelinated type of nerve, then at least 3 Ranvier nodes should be blocked before it clinically effective for the local anesthetic. Clear? So sodium channel, once if the local anesthetic block 1 or 2 sodium channel, the clinical effect will not be there. So if we give the local anesthetic, there is a increased amount, high amount of sodium channel should be blocked. Before there is a sequence. First there is a increased resting membrane potential. Then conduction will be delayed. Third one is the slope will be less stiffer, followed by amplitude will be decreased. Local anesthetic. After that, only after that, there is a complete conduction absence. For critical, 3 Ranvier nodes should be blocked for clinical effect. 3, at least 3. Now, what are the characteristics of this local anesthetic blockage? So, if we compare a thin nerve with a thick nerve, which one will be blocked prior? Thin nerve will be blocked priorly. Similarly, if we compare myelinated or non-myelinated, which one will be blocked priorly? Myelinated one will be, so thin myelinated will be blocked first in compared to the thick nerve or non-myelinated one. So, if pharmacodynamic action of local anesthetics came, not only the how it block the sodium channel, but also what are the factors that affects the local anesthetic, like diameter of the nerve. If it is less, it will be blocked earlier. If it is myelinated, it is blocked earlier. Similarly, if it is sensory type of nerve ending, it will be blocked earlier than the motor one. Sensory will be blocked earlier than the motor one. Similarly, if different type of A delta or C fiber, there are small diameter fibers, they block prior to the A alpha fibers. So, which one diameter is small, it will be blocked earlier. If it is myelinated, it will be blocked earlier. If it is sensory type, it is blocked earlier. If it is alpha A delta or C type, it is blocked earlier. So, these are the factors of the nerve which determines their onset of anesthesia, local anesthesia. So, if it in pharmacodynamic or mechanism of action, not only in a single line that it blocks the sodium channel, it's blocked the sodium channel in inactive state by binding in the inner surface, binding with the inner surface, number 1. It requires 3 Ranvier nodes to be blocked for clinically effective local anesthetic effect. Then the sequence, first resting membrane potential will be high, followed by the conduction, then less stiffer of this action potential height will be less, followed by total abolition of the conduction. Then which type of nerve is blocked earlier? In case of myelinated, small fiber, sensory fiber, alpha C, AC or alpha A or C, A delta or C is blocked earlier than the A alpha. Now, third one, which is determined the where the nerve supplies its, if it is proximally, then the fiber is more commonly in the periphery, whereas distally innervated nerve is more in the centrally located. This portion peripheral portion, mostly they supply proximally, whereas the central they go distally, they are proximal. So, local anesthetics, when given locally, the peripheral one will first anesthetize. So, the proximal portion of the nerve supply will be first interrupted, then the distal. Clear? If we go for the, bolo, বুঝতে অসুবিধা হচ্ছে? So, if we go for the cross section of a nerve, so there is a peripheral and there is a central portion. Peripheral portion, the nerve present in the peripheral portion, they supplied more proximally, whereas the central portion, they supplied distally. Anesthetic, they have which portion will be first anesthetized? Peripheral or central? Peripherally. So peripheral portion, if it is anesthetized, so supply will be less in the proximal part, because peripheral will be anesthetized, and later the central portion of the nerve anesthetized, so distal one is anesthetized lately. So what are the different types of use of this local anesthetics? Topical action. So if we have to give a incision only, suppose you have to give a intracostal drainage or a drain. So you have to apply this topically with a spray, or there may be infiltration. So this first one is topical use of local anesthetic. It can be sprayed with a local along with the adrenaline or in absence of the adrenaline. Next, in very close to peripheral nerve, it's called perineural block. Perineural, it is around the nerve. Around the nerve, it is perineural. Third one is inside a major trunk, nerve trunk. Example as major nerve. Which one is the thickest nerve? Sciatic. If you give it inside the sciatic blocks, that is inside the sciatic nerve. So there is a perineural, there is a nerve block, then epidural or subarachnoid space. We can give it epidural or subarachnoid space. And lastly, it can also give intravenously for extremity, operation of the extremities, upper extremity or lower extremity. We can give if the operation is less than 30 minutes. We can call it Bayer's block. So intravenous local anesthesia can be given. So ki bhabe dite hobe? Topically, number one perineural, then in the nerve trunk, then epidural, then subarachnoid, and lastly intravenous. লেখা যাবে এগুলো কি কি? টপিক্যাল, পেরিনিউরাল, নার্ভ ট্রাঙ্ক, এপিডুরাল, subarachnoid space, spinal anesthesia given in the subarachnoid space, and lastly intravenous, that is called Bayer's block. It is given primarily in the upper limb or lower limb operations where the operation is less than 30 minutes. Bayer's block. Clear? এর মধ্যে local MCQ short note can came from this portion. What are the different routes of giving local anesthetics? Here you can see the spinal block, which one is epidural block. Epidural, they doesn't penetrate the dura mater. Epidural just outside the dura mater, whereas if it is penetrate the dura mater, it is the spinal block. Similarly, if it is around the vertebral nerve, it is paravertebral one. বলা যাবে কোন কোন রুটে দেওয়া হয় লোকাল অ্যানাস্থেটিক? What is the difference between the spinal anesthesia and epidural anesthesia? In case of epidural anesthesia, it doesn't breach the dura mater, whereas if it is breach the dura mater, it goes into the arachnoid space. It is the spinal anesthesia. So there is a difference epidural and spinal. Why it is important? Because if the volume is much more in case of epidural one than spinal one. If we give the drug which is taken for the epidural and we breach the dura mater, it can cause total spinal anesthesia, and the low BP and patient will be may require intubation. So epidural and spinal anesthesia দুটো ডিফারেন্ট, কিভাবে ডিফারেন্ট? কোনটা পেনিট্রেট করে ডুরামাটা? স্পাইনাল. এপিডুরাল doesn't penetrate the dura mater. Vasoconstriction effect already discussed. Now sodium bicarbonate. If we give sodium bicarbonate for 1 or 2 ml, it maximizes the amount of drug and it is more lipid soluble. So action will be increased by adding the sodium bicarbonate. Similarly, if we repeatedly use local anesthetics, it can cause tachyphylaxis. Tachyphylaxis কাদের কোথায় পড়েছিলাম? Example, what is tachyphylaxis? হ্যাঁ, কোলিনার্জিক তো নয়, হ্যাঁ, টাইরামিন। এটা কি টাইপের, কি ড্রাগটা, কি ডাইরেক্ট না ইনডাইরেক্ট? ডাইরেক্ট আর ইনডাইরেক্টের মধ্যে পার্থক্য কি? সিম্প্যাথোমাইমেটিক। ডাইরেক্ট আর ইনডাইরেক্টের মধ্যে পার্থক্য কি? হ্যাঁ, অ্যাড্রিনার্জিক। অ্যাড্রিনার্জিকের ডাইরেক্ট আর ইনডাইরেক্ট অ্যাড্রিনার্জিক ড্রাগের মধ্যে পার্থক্য কি? হ্যাঁ, আরেকজন সেটা কোনটা? এক্সাম্পল কি তার? এক্ষুনি বললো tyramine is a type of sympathomimetic. When we use tyramine repeatedly in a short, we exhaust all the norepinephrine present in the synaptic vesicles. So there will be less action. Similarly, when we use local anesthetics repeatedly in a short duration, the extracellular acidosis causes tachyphylaxis. So tachyphylaxis for indirect type of adrenergic and tachyphylaxis for local anesthetics, the mechanism of action is different. In case of tachyphylaxis in indirect, there is a exertion of the noradrenaline in the sympathetic vesicles, whereas after giving repeated local anesthetics in a short time, the environment, extracellular environment became acidotic, so their action will be less. So তিনটে জিনিস, এক নম্বর হচ্ছে why it given along with vasoconstrictor like adrenaline. Number 2, why sodium bicarbonate increase its action, and third one is there is a tachyphylactic action for local anesthetics, and why. Toxicity. Majority 2 types of toxicity commonly. One is systemic, due to the systemic absorption, and due to the second one is neurotoxicity, direct neurotoxicity. 2 types of main toxicity. It is dose dependent. One is cardiotoxicity, one is direct neurotoxicity. These are the main common and severe side effects for local anesthetics. So in case of cardiac CVS, they can hamper the conductivity, the impulse generation. Similarly, in higher dose, they can also depress the myocardium. কি তাহলে হবে? ইম্পালস জেনারেশন লেস করলে কি করবে? ইম্পালস জেনারেশন কোথা থেকে হয়? এসএ নোড, সেটাকে ডিপ্রেস করলে ব্র্যাডিকার্ডিয়া হতে পারে। সিমিলারলি এবি নোড কন্ডাকশন যদি ডিলে করে ব্র্যাডিকার্ডিয়া হতে পারে। ইফ দেয়ার ইজ এ ডিপ্রেশন অফ দ্য মায়োকার্ডিয়াম, কি হবে? দেয়ার উইল বি হার্ট ফেলিওর। These are all effects of local anesthetic in CVS. Primarily, all these side effects is due to the blockage of sodium channel present in the heart. But in higher dose, these local anesthetics can also block the calcium channel. Clear? Local anesthetic, their primary action is due to the local anesthetic effect is due to the blockage of sodium channel কোথা থেকে তারপরে বলো extracellular or intracellular intracellular in which phase inactive phase not resting phase inactive phase and it is time dependent and voltage dependent এই কথাগুলো মনে রাখতে হবে এমসিকিউ তে আসবে রেগুলারলি যে টাইম ডিপেন্ডেন্ট ভোল্টেজ ডিপেন্ডেন্ট ইনঅ্যাক্টিভ স্টেজে ইনার সেলুলারে ব্লক করে সো not only they block the sodium channel in the present in the neurons they can also block the sodium channel that is present in the myocardium or heart by blocking that they can by blocking that they can reduce impulse generation reduce conduction reduce myometrial contraction by sodium channel in higher dose not only the sodium channel they can also block the calcium channel present in the cardiac system. Next one hematological specifically this is specifically for the pirdocane if it is given more than 10 milligram per kg they can causes methemoglobinemia due to its metabolite proto-oncogenes orthotoluidine for that they can causes methemoglobinemia and this for methemoglobinemia there is chocolate colored blood with a cyanosis patient may present with a cyanosis when we give methemoglobinemia give pirdocane more than 10 milligram per kg. Allergic reaction primarily with the esterase site. They metabolize to the p-aminobenzoic acid and they can cause allergic reaction. So cardiogenic, neurogenic, allergic and last one is allergic chhada hematological. So local anesthetic primarily explain wise and short note. Short note mechanism of action কি কি লিখতে হবে mechanism of action কিসের হবে হ্যাঁ mechanism